Hello, everyone. Thanks for your interest in my research. Here, I studied the characterization of intracellular Precore-derived proteins and their functions in HBV infection. HBV express Precore and core proteins using the highly overlapping coding sequence, but from two distinct templates. In contrast to the core protein, which form the capsid in the virion, the Precore protein is a precursor of secreted E antigen and the pre-C antigen. Several early studies have reported the intracellular Precore-derived proteins in the Precore transfected cells. However, the intracellular Precore-derived proteins in the HBV-infected cells have not been studied. And the functions of the intracellular Precore-derived proteins are poorly understood. Here, we characterize the Precore-derived proteins in the HBV-infected primary human hepatocytes. We include the wild-type HBV and the Precore-negative HBV G196A to infect the primary human hepatocyte and analyze the Precore-derived proteins in the cell lysate. We find there are several Precore-related species in the wild-type HBV, but absent in the Precore-negative HBV infected PHH. We tentatively assign this species as a P22, which is the intermediate of the Precore, and the non-processed Precore precursor P25, as well as their phosphorylated form. We also can detect this species in the HBV Precore transfected cells. To further confirm the assignment of the phosphorylation and the non-phosphorylated species, we did a lambda phosphatase treatment. Our result indicates that there are P22 and P25, as well as their phosphorylated forms in the HBV-infected hepatocyte and the Precore transfected cells. We further want to study the role of the HBV Precore protein in the context of the HBV infection. So we analyze the lysate of HBV-infected PHH. We find the Precore negative mutant should increase the levels of the core protein compared to the wild type. And also their capsid levels and the PGR levels also was more than the wild type. However, when we normalize the capsid levels to the core protein levels and our PGRA levels to the capsid levels, we find the Precore negative mutant showed no difference on the capsid assembly and the PGRA packaging compared to the wild type. This suggesting the Precore protein somehow affected the intracellular core protein expression. On the other hand, we analyzed the core DNA from the HBV-infected PHH. Consistent with the increased PGRA levels, the G196A also showed an increase of the single-stranded DNA, indicates it has increased DNA replication. However, we find the mature DNA species around on the top was accumulated in the wild-type cells, but not in the G196A infected cells. Interestingly, we find this mature species was released into the cell culture supernatant. This suggests the Precore protein might affect the secretion of DNA containing virions. Further, we analyze the viral culture supernatant. We analyze the cell culture supernatant using the native agar gel electrophoresis. We find that the Precore negative mutant should increase the levels of the DNA containing virion secretion, and also should increase the levels of the naked capsid release compared to the wild type. So this tells us the loss of the Precore associated with the increased levels of intracellular core protein and the secretion of DNA containing virions. Furthermore, we confirmed it using the Precore expressing construct and the HBV replicon co-transfection in the Hepatitis cells. We find with the increase of the P22, the P21 levels was decreased in the co-transfected cells, suggesting that intracellular Precore proteins may cause the degradation of the HBC. Interestingly, we find this decreased HBC levels correlate with the decreased levels of the PGRA and the DNA replication. This is suggesting the expression of the cytosolic P22 caused the degradation of the HBC, which further caused the decrease of the PGRA packaging and the DNA replication. We also find the P22 can form the complexes with the P21, And some of these complexes can package the pgRNA, which was resistant to the RNase treatment. This implies that the p22 and the p21 may form the chimeric capsid. On the other hand, we find the p22 alone can form some of the complexes that migrate slower than the capsid formed by the p21. However, this complexes was sensitive to the RNase treatment, indicates the p22 formed the complexes with the RNA. To further confirm the formation of the chimeric capsid, we resolve the cell lysate on the sucrose gradient. The p21 formed the capsid and sedimented in the fractions into the gradient. We find there are small amount of the p22 was co-sedimented in the fractions of the capsid. We further looked at these fractions under the TEM. We find the p22 alone can form the capsid-like particles, but this was very low efficient. On the other hand, we co-transfected the p22 with the HBV replicon. We find there are small amount of the p22 and the p21 co-sedimented into the fractions of the capsid. This suggesting that the p22 can form the chimeric capsid. On the other hand, the majority of the p22 and the p21 are sit on the top of the gradient, suggesting the p22 suppressed the capsid formation by HBC. On the other hand, we looked at these fractions on the native agar gel. We find confirmed that the p22 formed the chimeric capsid can package the pgRNA, and which was further confirmed by using the Precore specific antibody. This result demonstrates the intracellular p22 formed the chimeric capsid with the HBC. Next, we asked whether this chimeric capsid can be secreted out of the cells as the virion. So we analyzed the cell culture supernatant from transfected cells by the cesium chloride gradient. There are small amount of the p22 can be secreted at the lighter density fractions. However, the p22 and the p21, when they co-express, they are co-sedimented at the later fractions, which is the fractions of the naked capsid. When we have the HBV alone in the cell culture system, it has two peaks. One is a virion peak, and another is a naked capsid peak. We find the p22 and the HBV replicon co-transfection can suppress the release of the naked capsid and decrease the virion secretion, which was consistent with the result in the G196A and the wild-type HBV infected PHH. So we find the co-sedimentation of the p22 and the p21 in the virion fractions indicates the p21 and the p22 form the chimeric capsid. We further analyze it on the native agar gel electrophoresis. We find the virion peak was in the fraction 15 and 16, while the empty virion peak was in the fraction 14. We find the Precore signal co-migrate with the DNA signals and the empty virion signals, further confirming the formation of the chimeric capsid was secreted as the virion. So this result clearly suggests the cytosolic p22 was incorporated into the capsid and secreted in the virions. More interestingly, we find in the HBV infected primary human hepatocyte, in the cell culture supernatant, we resolved that by the cesium chloride gradient. We find in the virion fraction, there was a p22 and a p25 detected. More interestingly, we find in the HBV infected chimpanzee serum, there are also small amount of the p22 and the p25 could be detected. This suggests that the p22 and the p25 can be found in the supernatant of infected cells or the serum of infected animals, further suggesting the Precore protein were part of the HBV virus. As a summary, in this study, we identified the multiple Precore-derived proteins, including the non-phosphorylated and phosphorylated p22 and p25 in the HBV infected hepatocyte. And we also showing the Precore protein affected the HBC levels and HBV viral secretion. Furthermore, we demonstrated the P22 formed the chimeric capsid with HBC, which was secreted as a virus. At the end, I would like to thank for your attention, and I'm happy to take any of the questions.